This is our Project 3, Cell-Mediated Immunity. Chapter 5, T-cell mediated immunity. What are the functions of helper T cells and cytotoxic T cells? Helper T cells recognize antigens presented by antigen-presenting cells on MHC class I molecules. Once activated, they release cytokines, which are signaling molecules that communicate with other immune cells. These cytokines help activate B cells to produce antibodies, stimulate macrophages to destroy pathogens, and support the activation of cytotoxic T cells. Cytotoxic T cells. They recognize foreign antigens presented on MHC class I molecules. Once they identify a target, they release perforin and granzymes. Perforin creates pores in the target cell membrane, allowing granzymes to enter and trigger apoptosis or programmed cell death. So, what are the steps in the activation of T lymphocytes? The first step is antigen recognition. The activation process begins with the naive T cell encounters an antigen-presenting cell displaying a specific antigen. The T cell receptor recognizes the antigen MHC molecule complex, providing the first activation signal. So, step 2, cytokine secretion and receptor expression. After recognizing antigen, the T cell becomes activated and begins producing cytokines such as interleukin 2. At the same time, it releases expression of IL-2 receptors, making it more responsive to growth signals. Step 3 is proliferation. The IL-2 stimulates the activated T cells to rapidly divide, creating many identical copies of itself, and this is known as clonal expansion. So after this is step 4, these newly formed T cells are recognized into different types, including T effector cells and memory T cells that will help them fight infection and provide long-term immunity. Step 5 is the effector functions. Effector T cells travel to infected tissues and perform their immune functions. Helper T cells activate their immune cells, while cytotoxic T cells destroy affected cells. Memory T cells remain in the body and allow for a faster response in the same pathogen is encountered again. So what signals are needed to activate T lymphocytes? Signal 1, antigen recognition. So antigen recognition, the first signal occurs when the T cell receptor binds to the antigen presented by the MHC. The MHC? It's the MHC molecule by an antigen presenting cell. This provides antigen specificity. Signal 2, the second signal is a co-stimulatory interaction between CD8 and on a T cell and B cell molecules such as CD80 or CD86 on the antigen presenting cell. Without this signal, the T cell may become inactive or unresponsive. So survival. And signal 3 is cytokines. The third signal comes from cytokines released by surrounding cells. These cytokines guide the T cell's differentiation into specialized subsets and determine the type of immune response that develops.
Two signals are required for T cell activation. The first is going to be antigen recognition, when the TCR is going to recognize the peptide presented by the class II MHC molecule. We also see here that the CD4 co-receptor is going to bind and recognize the MHC molecule, which is going to help strengthen the signaling. And while the TCR itself is not able to transmit signals into the cell, it has to rely on the CD3 complex that contains signaling regions called ITAMs, and these are going to actually initiate the signal transduction. A second signal co-stimulation is provided when CD28 on the T cell is going to bind to the B7 on the APC, and this interaction is essential for the full activation of naive T cells. Without this, the cell can become inactive. And finally, adhesion molecules are going to help stabilize the interaction. We see this on LFA binding with ICAM. This is allowing the cells to remain in contact long enough for activation to occur. There are two major types of T cell mediated immune reactions. The first is going to involve CD4 helper T cells, and these are going to recognize microbes that have been ingested by phagocytes like macrophages, and after activation they are going to secrete cytokines that are going to activate more macrophages and recruit other leukocytes to destroy the microbes. THC1 cells are going to promote macrophage activation, while Th17 are going to promote the inflammation and recruitment of neutrophils. The second type is going to involve CD8 cytotoxic T lymphocytes, or CTLs, and these are going to recognize infected cells displaying microbial peptides on MHC class 1 molecules, and once activated are going to directly kill infected cells, eliminating the intracellular reservoir where microbes can replicate. Many infections are going to require the cooperation between these two mechanisms. CD4 cells are going to help activate the macrophages that destroy microbes trapped within vesicles, while the CD8 are going to eliminate the infected cells containing microbes in the cytoplasm. CD4 helper T cells are differentiated into specialized subsets based on the cytokines they are going to produce and the functions that they carry out. The first subset is Th1 cells, which produce interferon gamma, and the THC1 cells are going to activate macrophages through the classical macrophage activation pathway, and this is going to increase the production of the reactive oxygen species, nitrogen oxide, and the lysosomal enzymes, and this is going to enhance the killing of intracellular microbes, especially bacteria and viruses that are going to survive inside phagocytes. The second subset is Th2 cells, which produce interleukin 4, 5, and 13. These cytokines being responsible for the production of IgE, mast cell activation, eosinophil activation, which are going to directly fight against these helminth parasites. And they are also going to promote the alternative macrophage activation, which is associated with tissue repair, wound healing, and fibrosis. The third subset is going to be Th17 cells, which produce interleukin 17 and 22. Interleukin 17 is going to stimulate the release of chemokines and inflammatory cytokines that are going to recruit neutrophils and monocytes to sites of infection. IL-22 is going to promote the production of antimicrobial peptides and helps maintain the overall integrity of the epithelial barrier and tissue repair. These are responsible for protection against extracellular bacteria and fungi. And lastly, the T follicular helper T cells are going to help with the activation of B cells and support the production of antibodies. So overall, I went through the discussion of how activated T cells are going to recognize targets and coordinate specialized immune responses. And my source was going to be just the textbook for the course.
Mechanisms by which T cells activate macrophages. There are two main mechanisms by which T cells can activate macrophages. The first one being the M1. It is induced by Th1 and are activated through the secretion of INF gamma or interferon gamma, which is the most potent macrophage activating cytokine known. These macrophages are pro-inflammatory phagocytes that kill ingested microbes. The second being M2, which is induced by the Th2 cell and are derived by interleukin 4 and interleukin 13. These macrophages serve as tissue repair and are not microbial killing. Responses of macrophages that result in the killing of ingested microbes. Once the microbe is ingested by the macrophage, it represents it to the CD4 T helper cell via MHC2 peptide complex. Following this, the CD4 T helper lymphocyte secretes cytokines such as INF gamma or interferon gamma that command the killing of the microbes in the phagolysosome. This is also the classical macrophage activation. How CD8 cytotoxic lymphocytes kill cells infected with viruses. A CD8 cytotoxic T lymphocyte first recognizes an MHC1 peptide complex on an infected cell with its T cell receptor, and then forms a tight conjugate with a binding of LFA1 and ICAM1. Once stable, it releases perforin, which helps the delivery of granzymes into the infected cell's cytosol. This then induces apoptosis,